Assalamualaikum, this is my project GeneLens. I have used a distributed DNA analysis and mutation-based disease prediction system to perform the task using protein folding concepts. Later on, the main objective of the program is to use Clyde to sense the DNA sequence, and it performs the multiple computations and tasks to detect the diseases and DNA sequence alignments and scores, and that information is sent to client, and there some visualizations and report generation is done. Now I will be moving forward to show the main output and lookup of the project testing. So as to make my project more real and more efficient and visible for the GeneLens or bioinformatics technology and systems, I created and decided to use a database for genes and mutations, where I have added two tables where there is a healthy gene database and there is mutation gene tables. I have used a real snippet and a more authentic sequences of characters in my database, so the results are shown more accurately. So let's start with the main system, and here is my client side where the sample is sent from client to server, and server is basically the main load balancer or the distributor, which is basically dividing the traffic or the load from server itself to different worker nodes. So it has efficiency and more capability of doing a large number of samples of DNA sequences at a single time. That's why I've also integrated the cloud system, which I have for now. I have also done the setup for now. Here, if I enter the sample ID, this is for making a generation report. Uh, I would give it name as sample one, and it will then ask for the DNA sequence. And if I give it DNA sequence, we will be able to show get the DNA analysis. As I um, press enter and make it analyze the output, it went from server side after receiving the DNA sequence from server. It went to the worker node. As these worker nodes were free, it calculated and did all the computation of DNA analysis. And after the DNA analysis were performed, it got received at the client side. It showed the analysis, the score system, the aligned input and gene. Basically, it is showing the few of the mutations with the major chunk which was matched and how many updations, insertion, or deletions are required according to the database it had. And if you are able to see there is still no mutation, um, not much mutation in the if we see with the respect of database, and it saved the file with date and time recorded and saved it in a text file, as well as showed a data visualization in which we are able to see that these are the mutations which were happened, but this was the part where this was a full match. And if we move to the local files, it saved a text file with the sample on the client side. So it had better visualization and uh, better user interface for client side. I have another screenshot where it is showing a disease predicted and with its mutation and alignment score, which is also saved in an sample file in text file at client location.